Okay, so this is the rest of lecture 23 prep. And we had just finished talking about how the organizer properties change over time. That early organizer provides anterior information, later is more posterior information. Um, and so in the early gastrula, the cells of the dorsal lip will, as they're moving around, they give rise to anterior endoderm. Let me get my pen here. We'll do green, which is going to form the anterior end of the gut. Precordial plate mesoderm, which is the name of what's going to form the head. Precordial means it's before the notochord. So you've got the notochord, and then there's some mesoderm here that's ahead of it on the anterior side that's going to participate in the formation of the head, the mesodermal parts, the muscles and so on, and then the notochord. And goosecoid is expressed in all the cells of the early organizer, and it's required for the formation of the head. So it's required for the formation of the the precordial plate mesoderm, which is actually going to form the head. But the organizer is also divided. So if you look at sort of the dorsal lip of the blastopore, where the blastopore is starting to form, the more vegetal side of it is expressing OTX2 and LIM1, which are activated by VEG-T, or old friend VEG-T that we've been talking about for a couple of lectures, and is shown on the diagram in the next slide. And then on the dorsal side, it expresses NOT, and NOT is required for notochord development. And that's shown also on the next slide. So let's just take a look at the diagram, because I find the diagrams are better for explanation. So here's our early, very early gastrula. Just the tiniest little bit of the blastopore is starting to develop. Now, as the cells inside move around and in, you have these ones that are called the leading edge cells. And they're going to move in, and they kind of migrate pretty actively across the top of the blastocele cavity, and they're going to move all the way around, and they're going to end up right up in here, and they're going to form anterior endoderm, which is the beginning of the gut. Now, because of cell adhesion, so connections between the rest of the cells are kind of pulled along on this journey. And the cells, so if this is the beginning of the blastopore formation, so this is the vegetal side of the organizer, and this is the dorsal side of the organizer. And the ones on the interior here of the organizer, the more vegetal ones, are expressing these two transcription factors, and these are required for the formation of the precordial mesoderm. So that's the head mesoderm. And then above that is going to be the anterior neural plate. And so these cells are going to participate also in the formation of the brain and that sort of structure, the anterior head neural tube. And then the notochord is is formed from some of these cells that move in from the more dorsal side of the organizer. And notice that there's a ton of these cells. And it's not just that they're rearranged. They are also, it's really hard to draw these diagrams, but part of it is because the cells are still also dividing as movement is occurring, and so on. So there's many things happening simultaneously. Okay, so these are going to give rise to the notochord, and then of course the neural plate develops above them, and the neural tube is going to develop as well. Okay, and the mesoderm that hasn't yet formed the notochord but is going to is just called the chorda mesoderm. I shouldn't have crossed out notochord, but I was just trying to emphasize this name that leads to that structure. Okay, so that's kind of where things are, and these are the only factors for this kind of thing that you need to remember. Okay. Recall that the organizer secretes a lot of antagonists of BMP and Wnt signaling. And so that means the levels of these kinds of signaling in the organizer are really low. Okay. Now we haven't talked about Cerberus yet, but it actually specifies anterior endoderm. So it's being expressed in the organizer, and these are the cells that are going to form the anterior endoderm. And so I'm just going to add this on here, Cerberus. Okay, that's not spelled right. Cerberus. Okay. And it antagonizes BMP, Wnt, and nodal, right? So these are BMP, nodal, TGF-beta family members, and Wnt signal. Now, early on, very, very early in development, you know, in the early, before the blastula is even fully formed, we have a dorsal to ventral gradient of Wnt signaling, which specifies dorsal versus ventral fates. Remember that the organizer, after it forms in the early, later in the blastula stage, it's secreting inhibitors of Wnt, and so the gradient is kind of, it's not as high a gradient, but it's going in the opposite direction now because there's more Wnt signaling on the ventral side at this point than there is on the dorsal side. So there, something to think about, and just we're only going to look at these 3 points, 3 time points, is that at different times there's slightly different Wnt signals. There's multiple. You noticed X Wnt 11 and X Wnt 5a. That means that there's a Wnt 1, 2, 3, 4, etc. There's other Wnt signals as well. Wnt 8 is one of the mesoderm specific ones at this stage. So there's all these different Wnts, and they have slightly different targets, and they're happening, the gradients, at different times. So the dorsal ventral, and then ventral dorsal, and then posterior anterior is another Wnt gradient. And so I just want to make sure that that makes sense. So at the very beginning of development, we have this dorsal to ventral Wnt signaling gradient. As, remember, the organizer is developing, then there are these inhibitors that make the gradient kind of, you know, work somewhat the other way around. And these are the signals. So early on, nodal related is one of the targets of Wnt signaling on the dorsal side. You can leave out Dickkopf, and just note that Cerberus, Noggin, and Cordon are also targets of this early signaling, and the signaling is really strong here on the dorsal side, and that's why then there is this feedback kind of thing that happens, where Cerberus, Noggin, Cordon are activated by Wnt, and then they are part of the inhibitors of Wnt and BMP later in development as well. So Noggin and Cordon inhibiting BMP, Cerberus inhibiting Wnt signaling right in this region. So this area kind of clears of those kinds of signals. Okay. Later in development, in the very late blastula, early gastrula, the cells start moving around, and the gradient takes on a different role. Now you have a Wnt gradient that is higher for the posterior and lower for the anterior. So there's a posterior to anterior gradient. And notice, I don't think I'll ask you to memorize any of these, but just notice that these are all really different targets, and they're important for the specification of ventral posterior mesoderm. And so they're doing different things at different times. If you remember the targets that I've noted here, and then just note that the later ones are different, we will probably see these genes at a later time in other contexts, but for now this is all you need to know. Key point here, different timing and different targets, and so Wnt signaling does different things at different points during the developmental process, and they are different Wnt's, and they have different target genes. Okay. And so here are some experiments. If you inhibit Wnt and BMP in the ventral region of an early gastrula, you can induce a second head. So the organizer region is required for induction of head, and if you recreate those conditions in an area that should have high levels of both of these signals and create a little cleared area where those signals are not taking place, you get a second head induction, just the way you would if you did an extra cortical rotation or you introduced new Coupe center molecules in that region and so on. And it turns out that inhibiting both BMP and nodal can do this as well. And remember that at this time, nodal is one of the targets of Wnt, right? So you can either inhibit Wnt and BMP or one of the targets, the most important target of Wnt, as well as BMP, and you get the same result. And so this is just a little bit of the evidence, but this and quite a bit of other observations have led to this model, the two gradient model, where you've basically superimposed an anterior-posterior gradient of Wnt and a dorsal-ventral gradient of BMP. These two gradients are functioning early to late gastrulation. So this is, again, another time we've got these two different gradients. It's being set up during the blastula stage, but it really has its effect in gastrulation. But when we look at the actual gradient images, they show it usually as these nerulas, because during gastrulation, you've got cells moving, from where it's dorsal around, and suddenly the dorsal is up here, and then it's just because of the way the cells are moving around, it's easier to understand if you look at the finished product, which is the little nerula embryo with the BMP gradient, with the ventral having large amounts and the dorsal having very little or none, and then the posterior has a large amount of Wnt and the anterior very little. And so any place in this embryo, you can tell whether the cells are more ventral or more dorsal, because the ventral ones are going to have high levels of BMP and the dorsal ones are going to have low levels. And then the more posterior the cells are, the more Wnt signal they're going to have. And so, of course, there's intermediates, right? There's intermediate. So if you're halfway along, you have about half as much of each. If you're halfway down the side here, you have, half as much. So there's intermediates too, but these are the extremes. So the anterior dorsal most part of the nerula is low for both. That's what the lowercase and non-bolded means. The posterior and ventral has the highest amount of both, so that's what the uppercase and bolded means. And then on the posterior side, you have lots of Wnt signal but very little BMP, and the other way around for anterior ventral, very little Wnt but high levels of BMP. And so this is just this is my sort of schematic. I like to make like little tables and things like that just organizes the information so that I can remember how something like this works. And I'll probably ask questions about this in the class to try to ensure that it's understood. Okay, so that's sort of the general anterior, posterior, dorsal, ventral. So when we look at the early gastrulas, the presumptive neural ectoderm, the stuff that's going to form the neural ectoderm, is found just a little bit above, of course on the outside, but just a little bit above the organizer. And the presumptive epidermis is kind of across the embryo, so it's more on the ventral side, and there's lots of that. If you were to take and somehow label these cells with a dye or a fluorescence or something, and you looked at the normal development, you would see that these are going to form neural tissue. The whole neural region is sort of outlined this way. Not very neat, but the cells that you've labeled are going to show up as part of this neural system. And the same here, if you were to label these cells and then observe them, follow them through development, you would see them showing up in the epidermis and not in the neural cells. And of course, if you just label this little patch of cells, a lot of the epidermis is not going to be labeled. So this just, shows a subset of the cells that you would that are epidermal. Okay, so now what happens if we take presumptive, say, epidermal tissue and transplant it into a region that is supposed to form the neural tissue? Remember how this kind of experiment would have to be done. You want to be able to follow the transplanted cells inside the host cells to find out what happens to those transplanted cells. And I may ask about that in the class, so think about that in advance so you're kind of prepared for that kind of question. But you can see, in this case, the transplanted cells that were supposed to form epidermis, when you move them into the region where neural tube normally, or neural tissue, because it's not all neural tube, but where the neural tissue is going to form, it takes on that fate. Okay, so if you think about this is how they I think we're thinking at the time, is you've got a relatively simple tissue, which is epidermis, and then you've got this much more complicated tissue. I mean, think of the nervous system, like your whole body is basically run by your nervous system, right? And so it's more complex tissue, and the idea was there's probably a signal that induces, and here's your organizer right down here, so probably maybe it's coming from the organizer, and it's going to induce neural tissue in the area directly above it. So when you look at those fate maps and you remember, you had the notochord mesoderm and sort of right where the organizer is, and then you had the band of neural tissue above that, and then the band of epidermal tissue further away. So you might imagine signals coming from that organizer to provide specific information on forming neural tissue. So there was a huge search for the neuralizing signal. Okay, and most labs that were studying this, were operating under this kind of assumption. So there's a large amount of research where they were trying to identify the factor or factors, because it's complicated. It's complicated. So we would expect, there might be multiple signals and so on. And so here was a little bit of some of the early work. So the early work showed that if you, even if you had dead cells from the organizer, and you cultured them with epidermal ectoderm, you could induce it to form neural tissue. So that was pretty promising, because that would suggest there's something in this organizer that can induce neural tissue. And so there's tons of different signals, and there were many different protein factors that were tested. And so you could take animal cap cells, or presumptive, say, epidermal cells, and then culture them with different kinds of signaling molecules and so on, and see if they induced neural fates. And so many of them did. The vast majority of the factors and treatments that they tried seemed to be able to induce this neural fate. So it was kind of confusing, because you wouldn't expect, like, every single, well, not every single, but the vast majority, you would not expect so many different factors to be sort of neural inducers, right? So subsequent work showed that when you took presumptive ectoderm, epidermal ectoderm, and just cultured it on its own without any treatments at all, not always, but it would often transform into presumptive neural tissue. So this didn't seem like the right organism to be trying to figure this out. So they switched back to frog embryos, and the culture of the epidermal ectoderm alone was not so easily transforming, like just spontaneously, into neural tissue. But here's a kind of a key observation. They found that if you disaggregate the cells, so embryonic cells are quite sticky, there's lots of cell adhesion and molecules and so on holding them together. And so there's ways of disrupting them without actually damaging the cells. So the cells are all separated from each other. And then if you withdraw that treatment, they will reaggregate, they'll stick back together. And if you do that, they will then differentiate as neural tissue. So this is really interesting because you wouldn't think that just separating the cells and then sort of letting them reconnect with each other would do this. So the question is, well, what's happening? Are there gene expression changes when this happens? And if so, those are the ones to start thinking about as our potential neural induction system. So these are the three key observations. The first is that when you do that disaggregation, the separation and then the rejoining of the cells, you end up with downregulation of BMP signals in those cells. So they're not signaling to each other. Noggin, which is a BMP antagonist, we know it inhibits the signaling from BMP, will induce neural fates in explanted tissue. Now, this alone isn't enough, but when you add it to the other pieces of evidence, it's compelling. Because remember, lots of different molecules, when tested, could induce the neural fates. So it's the combination of that information with the other two. And then when you, if you do careful tracing and you observe the formation of the neural plate, you can document that BMP expression is downregulated or lost in the actual embryo as this process takes place. So this suggests that it's not actually a signal that's activating neural expression, but in fact it's the reduction of or loss of BMP expression, the downregulation of it to allow neural tissue to develop, which would suggest that BMP is basically preventing neural fate. And so you have to repress the repressor in order to get neural fate. It's kind of interesting. It would suggest that this is a default and that something has to happen to prevent it from happening everywhere. And then you have to just withdraw the repressor in certain locations to allow neural development. It's almost like a double negative, right? And so they tested this by doing an experiment where they used RNAi, I believe, or possibly an antibody-based method to deplete embryos of BMP and to see if that was the actual trigger. Okay, so basically this is the experiment. Here's a normal pair of nerulas. This one has been stained with Laxed, and SOX2 is a gene that's expressed in neural tissue. Cytokeratin is a gene that's expressed only in epidermal tissue. So you can see under normal circumstances the developing neural tube and the neural folds and so on showing up blue, and the rest of the embryo is white. And then almost the reverse of that, all the epidermal tissue is blue, and there's a white region which is the nervous system, because the nervous system doesn't express keratin. So there we are. These two genes are markers, and there's certain ones that are used commonly as markers of particular tissue types. If you look at embryos that have had BMP depleted, when you get rid of BMP expression across the whole embryo, the entire embryo, the ectoderm at least, is specified as neural tissue, and that's why you get this blue color everywhere. And when you stain that same embryo, well, a different embryo, but with the same treatment, it's not the same embryo, the same individual, then you basically get no epidermis, or maybe a teeny bit of expression around the edges, but the vast majority of this has been specified as neural tissue, and so therefore there's no keratin expression. So this is really nice evidence for the role of BMP. And this is just a schematic that shows that. So this shows BMP4 over the entire embryo, the organizer region forming where the BMP antagonists are found. And then these antagonists are actually inhibiting. So we've already talked about cordon inhibiting the signaling and confining BMP4 to the more ventral part of the mesoderm. This also takes place endodermally as well. And then the prospective neural fate is again, we have these inhibitors, and they are basically preventing BMP signaling in the most dorsal part of the ectoderm, and that is leading to neural development. So same sort of process, the same subset of molecules working via the same mechanisms, and in the ectoderm, mesoderm, and endoderm, you're going to have something similar, which is the promotion of more dorsal fates and confining the more ventral fates to the actual ventral side of the embryo. So importantly, the neural induction, there is what we call redundancy or backup. So BMP4 promotes epidermal fates and by basically blocking neural development. Because if you get rid of BMP, then all of this is going to be specified as neural. And there's a whole host. There's 4 different molecules that are inhibiting BMP. Cerberus also inhibits Wnt. Okay, so it's not just acting on the one. But these guys are blocking BMP signaling to allow the neural development. We're only focusing on this part right here. And it turns out you can't just knock out follistatin, for example, and get the neural ectoderm not forming. You can't just knock out cordon and then get the neural ectoderm not forming. You need to see a combination of them knocked out because it's the formation of a nervous system is a very important part of development. And if you don't have a functional nervous system, you don't have a functional organism, right? And so there is a lot of backup. And so if in order to see a defect in neural patterning, you have to see cordon and noggin both knocked out, plus follistatin or plus Cerberus. So 3 of the 4 molecules have to be inhibited or inactivated in order to actually see that the neural ectoderm then doesn't form properly. There's also a positive signal, which is FGF, and that's positive to promote neural development. Okay, so you do have an activating factor as well. But the most important one is the inhibition of BMP4. Okay, so this is a quick summary. And basically in the animal cap cells, the epidermal fate is through BMP. The prospective neural fate has the inhibition of BMP and the active role of FGF as a positive. And actually nodal as well, which we didn't really talk about. But in the animal cells, we talked about this previously in a different lecture, that nodal function is inhibited by ectodermin. Basically, ectodermin is antagonizing nodal function in the in specification of mesoderm, right? So nodal is basically in the equatorial cells specifying mesoderm except the head mesoderm. Everything, BMP, Wnt, and nodal in the organizer region are inhibited, and they are going to form the head mesoderm. Some nodal is required for notochord formation, but the other 2 are inhibited. And then prospective muscles, which is still intermediate mesoderm, FGF and nodal together are both promoting that. And then on the vegetal side, prospective endodermal fate requires nodal and the inhibition of these. Now you can go through as a study guide or a study method, and we haven't really talked about FGF very much. It's kind of an afterthought. But you know what some of the inhibitory molecules are in here. And you might want to just remind yourself of what those are from previous lectures. For example, you can talk about sort of ectodermin here. because this is inhibiting the mesodermal fates to allow for an ectoderm, and then whether it's BMP present for prospective epidermal fate or BMP absent for neural fate, right? That's kind of how we're thinking about this. Okay, I think I'm going to stop because I think it would be good to just, the last few slides, yeah, I'll just do another one because it's dragging on a bit long. So we'll pick this up here next time.